We're going to talk a little bit about just chromosomes for a while, a few of the upcoming lectures. And shown here, this is an image of chromosomes from a fruit fly, similar to what we do in the laboratory. And you can see there some banding patterns. The difference in the banding, which is also contrasted by blue and yellow, the bands on here, the thicker, darker parts, those are the regions where the DNA is tightly packed, a little more tightly packed than the looser regions, like here is a little looser. This is one of the features of chromosomes. So it's basically packed up DNA and allows the cell to have this compact DNA for moving around mostly. So one thing I like to emphasize during this topic is that we don't always see chromosomes looking like this. A lot of times chromosomes are quite loosely arranged. They get packed up tightly under certain circumstances. So just kind of backtracking a little bit, what is a chromosome? We talked about some molecular genetics already. So we talked about DNA being a molecule, and chromosomes are really long stretches of DNA. So millions of nucleotides that are double-stranded DNA all packed together so tightly that basically we can take all these nucleotides worth of information and you can actually visualize this with a high-powered microscope. We've also talked early on, we talked about life cycles and how alleles are transmitted, and just a little more thorough coverage of that topic right now. So this is a human, what's called a karyotype. So the arrangement of chromosomes that you would find in a human. As we talked before, the chromosomes are paired and the pairs are homologous, meaning that they're the same size and they have the same centromere position and they contain the same genetic information, the same content of genes, although they can have different versions of those genes. So the coloring on here is an artificial coloration where they put a DNA probe into different parts of the genome, and they basically put probes that come out yellow so that they attach to the DNA, the distinctive DNA that's in this portion of chromosome 2, and so on. So a person could get this karyotype and line these up as they have done here. What you see, this is not how the chromosomes would arrange in a cell. They just be kind of randomly distributed. This is the result of somebody taking a digital image and cutting and pasting the various chromosomes so that they line up in homologous pairs. And then also, as we saw in the sex inheritance lecture, the individual here would be a male, showing X and Y chromosomes with a different shape, even though they are sort of technically a homologous pair of chromosomes. All right, so where do we find chromosomes? Well, think of that on a cellular scale. Depends on the DNA you're looking for, and it depends on the organism you're looking at. So we'll break it down between bacteria and eukaryotes. We've seen a few reasons why these kinds of organisms differ. Another one is how their chromosomes are put together. So bacteria have, in some cases, just one chromosome, sometimes more than one chromosome, but it's loose and inside of the cell. So bacterial cells are much smaller. The scale is not quite the same on the eukaryotes and the bacteria. Very small cells, and the cytoplasm doesn't have any organelles. That means the DNA is just out there in the cytoplasm. In a eukaryotic cell, we often think of the DNA being in the nucleus, and certainly a lot of DNA is in the nucleus. That's the hallmark of being a eukaryote, is to have this nucleus with DNA inside. And just a little note about the other places you can find DNA. These eukaryotic cells also have a little bit of DNA in their mitochondria. And if you're a plant or photosynthetic eukaryote, you have a little bit of DNA also in the chloroplast. So it's kind of in several places, and sometimes it's important to know where you can find those. So just kind of thinking about the different organisms, and in these organisms where you might be looking for a gene. Fruit flies are eukaryotes, so you would have nuclear DNA, and they also have mitochondrial DNA. Tomato plants, nuclear DNA, mitochondrial DNA, and also chloroplast DNA. And then this bacteria, as we saw in the last image, just have its DNA, its chromosomes and genes out in the cytoplasm. So a little more detail here, showing you some relative size also. So start off with a rather simple organism. This is the bacterium that causes tuberculosis, Mycobacterium tuberculosis. And it's got a genome. This whole genome is 4.4 million base pairs. So bacterial chromosomes are circles. So they don't really have a beginning or an end. It's a nice way to have stability to the DNA. But it also, they can have just one of these sometimes in the whole organism. So this is the distinctive circular genome of this organism. And these little arrows on here kind of give you a feel for the scale of the genes. Each of these little arrows is a gene that belongs to this organism. So not a terribly large number of genes, but of course this is a simpler organism. So moving up a little bit in complexity, our fruit flies down at the bottom are the structures of the nuclear chromosomes. So chromosomes 1 through 4 in fruit flies, they have only 4 nuclear chromosomes. The first one's called X. The second chromosome, 2 and 3, have these left arms and right arms, so 2L and 2R, left and right arms of chromosome 2. Same thing, chromosome 3, left arm, right arm. So the genes that we study in fruit flies, they should be located in most cases on one of these 3 kinds of chromosomes. Chromosome 4 is very tiny, and I don't think I've encountered a gene that's located on chromosome 4 in fruit flies. Fruit flies also circular chromosome, and that belongs to their mitochondrion. So a little bit of trivia that the mitochondria has its ancient origin in bacteria. So the mitochondrion is basically a consumed bacterial cell that ends up doing cellular respiration for eukaryotes. A little bit of relative comparison in genome size. We saw the tuberculosis genome was 4.4 million base pairs. Fruit fly here, we're looking at 123 million base pairs. So in a lot of cases, the complexity of the organism tracks with the number of genes, the number of nucleotides of DNA, and then moving up to a more complex organism, humans, about 3 billion base pairs. Also number of genes, 17,000, 30,000, moving our way up through animals. Plants, as I mentioned, lots of variation in the chromosome number of plants. This is tomato, and it's got 12 chromosomes shown here. Also a mitochondrial genome shown here, also chloroplast genome shown here. So all told, more genetic information than we saw in the fruit fly, and a little bit less than what we see in the humans. So sometimes what we think of as a complex organism might not always track with the evidence for its genetic complexity. So looking more at the human mitochondrion, we talked in the sex inheritance topic about mitochondrial disease. And so this relates to the genes that are actually located on the mitochondrial genome. So if a person has a significant mutation in one of these, they can be defective in a very central process to life. So you'll see a lot of the genes on here. I put the abbreviations that are on the diagram and spelled out some of these. NADH dehydrogenase, cytochrome, these are molecules involved in cellular respiration. So the mitochondrion is the location for cellular respiration, and also a lot of the genes for doing that process are in the mitochondrion. And then it also has its own transcription translation system. So tRNAs, rRNAs, still present in the mitochondrion genome. But overall, not very big, 37 genes. Almost seems like it's hardly worth the time. 16,500 base pairs, very small. So the streamlined, tiny genome for the mitochondrion in animals, that's kind of similar to what we see in, so on the order of thousands for mitochondria and chloroplast genomes. And just some examples, I'm coming at this from familiarity with plants, so I know about some rather extreme plant examples. These are variation in the genome size, so number of nucleotides of genetic information in the genome. On the left is a plant called Genlisea. 61 million base pairs, and on the right, a plant with 149 billion base pairs making up its genome. It's a little bit unfair. Sometimes plants will have extra copies of their chromosomes in a situation called polyploidy, which is not uncommon in plants and can sometimes kind of inflate this number. So it's not necessarily the number of unique contributions to this genome is so much bigger. It might just have an additional number of chromosomes. So variation in the genome size, and then also variation in chromosome number. The smallest number of chromosomes in an organism is this 2n equals 2 of an ant. And I believe the largest maybe that I could find anyway, as this 2n is 1,260 in this rather inconspicuous fern plant here. Somebody had to count those. So 1,260 chromosomes in a diploid fern plant. So as I mentioned, the bacterial chromosome is kind of ancestrally way back in time. Bacteria would have been the origin for mitochondria and also the origin, a separate origin for chloroplasts. We see lots of good evidence for that. So you can relate the structures of the bacterial chromosomes to the structures of the mitochondrial, the chloroplast chromosome. They also have, similar shape, similar size, and this double membrane here on the chloroplast of mitochondria is thought to be a remnant, a leftover of the fact that that inner membrane was the outer membrane of the bacterium that got consumed. And the outer membrane is the sac that it was consumed into. So kind of a cool parallel there across very, very distant organisms. So if we want to look at copying chromosomes, it's probably easiest to start with something like a bacterium that has a simpler set of chromosomes. So if you have just one circular chromosome, we've seen it drawn as circles, but of course in 3 dimensions in life, the DNA doesn't hold a perfectly circular shape, so it's going to fold back over on itself a little bit. So it's kind of a floppy string kind of molecule inside of the cell. Takes up a lot of the space in the cell. These are really streamlined organisms. All right, so the process of copying a cell also entails copying the DNA. So if you're going to make one cell into 2, then each cell that results is going to have a copy of the genome. So if you start with one genome in one cell, first step is to make 2 genomes. And then the next step is to make 2 cells, where each cell gets a copy of the genome. So the genetic result after this cell division is you should have 2 cells with exactly the same DNA as the cell you started with. So I mentioned that chromosomes are tightly packed DNA. This is the kind of the scales of packing DNA. I'm not too concerned that you know really anything about the details on here. You probably had it before and you may have remembered it from there. Basically, this is the double-stranded DNA molecule. And at the first scale of organization, it wraps up around these little proteins called histones. And a group of histones together with their DNA wrapped around them, that's a nucleosome. So they kind of look like beads on a string. And that allows these molecules to pack up nice organized fashion. So just sort of like a phone cord or electrical cord that you want to keep track of. It's important to make sure you don't get it too tangled. So this is like a fine molecular scale process where these wrappings of molecules don't get themselves tangled up. And not only that, but the genes that are contained on this DNA can be accessed using molecules that basically recognize and know where to access them. So basically it adds up in complexity across larger scales, 10 nanometer, 30 nanometers, 700 nanometers, 1400 nanometers, and it's packed up. And then the packed up bits are packed up also. And eventually they get big enough that we could see them under a high-powered microscope. And this is kind of the characteristic chromosome shape. And as we'll see, this chromosome shape is not always present, but it's really there during the process when the DNA is being moved around. So a few examples of how the DNA might look if you encountered cells. On the left hand here, this is what we typically would see inside of a cell. So if you want to see these nicely packed up chromosomes, people typically have to look for cells that are in a special stage of division. So you can get cells and either induce them to do mitosis or maybe catch them in the act if they're a certain kind of cell. Cells that are actively dividing get you more likely to see this kind of chromosome shape. If they're not actively dividing, they're more often just loose DNA. And what you see here, the blue stains the DNA, and the circle is the basic form of the nucleus. So you can't really make out any individual chromosome, but sort of rest assured that they are in there. And when it comes time to move them around, they could be packed up into chromosomes like this. So relaxed DNA, packed up DNA during the process of mitosis or division of a single cell into two cells. And then over here during the process of meiosis, dividing chromosomes from one starting cell into 4 final cells that will become gametes. So as I mentioned, to see chromosomes that are packed up, it helps to look at a place where cell division happens pretty rapidly. And this is one such place. This is an onion root tip. So one of the ways you can do this in the laboratory is you just put like a supermarket onion in some water and it starts growing roots. And as you, if you catch the tip of that root as it's growing, the growing entails a lot of cell division, and you can catch a lot of these cells in the middle of doing their chromosome division. So again, a couple examples. The majority of cells in here are not showing their chromosomes packed up, but a couple of them are. Also shows you that things are still kind of floppy, even though they're packaged up as chromosomes. All right.